Hello, good morning, dear students. I am Dr. Sunil Kumar Chaudhary, your bio teacher. In standard 9, you have to study a number of chapters. The first chapter is the cell. You know, cell is the fundamental structure, that is the structural and functional unit of life. In bio, we study different types of organisms. It may be the unicellular or multicellular. But without knowing cell, you are not able to understand the living organism. Whatever the features we have for being a living organism, all these features found in cell. When we study cell, it has different characteristics. And in bio, a separate branch develops. So overall, the study of cell, its structure and functions, that comes under a new branch of bioscience, and that is cytology. Cytes in itself, C-Y-T-E-S, cytes, that means cells. Up to now, we have gathered lots of knowledge about the cell. But the cell started its journey after its discovery. And the discovery of cell is also started with the discovery of microscope. If you go through the different aspects of the cell, you will find how the microscopes help in understanding the structure and function of the cell. The discovery of cell, that is, from that time, we started knowing the cell. The cell was discovered first of all by Robert Hooke when he observed a very fine slice of cork. Cork is a non-living material obtained from the bark of a plant, Quercus subericus. The cork that is almost dead, full of suberin. When the Robert Hooke observed the fine slice of cork under the microscope developed by himself. So self-made compound microscope. What is compound microscope? When a microscope having more than one lens, biconvex lens. The biconvex lens is the magnifying glass. When we observe anything under the lens, biconvex lens, we see anything in magnified form. That means even the very small thing appears large sized. So from this point, the study of cell started, and later on, a new branch of biology developed, and that developed into the cytology. A number of cytologists contributed a lot, but the microscope started its journey with the discovery of lenses by A.V. Leeuwenhoek, Antony von Leeuwenhoek. He was the first person who developed a number of lenses and saw bacterial cell, a sperm cell, and so on. But this discovery, it led by the Robert Hooke. Later, a number of biologists came forward, and then we know a lot of things about it. So contribution of Leeuwenhoek, Robert Hooke is great. At that time, there was no sufficient knowledge. Robert Hooke observed only the cell boundary, almost dead cell. Robert Hooke gave the term for that outer boundary of the cell. He observed the many compartments of the corks, and so he gave the name cell. Cell derived from the word cellula or cella. So cell is very important one because it is the cell which are the structural and functional unit of any living organism. After this, you see another slide, how the different types of the microscope. The first slide, that is the self-made microscope by Robert Hooke. Second, the modern microscope. It is light microscope. That means for observing any sort of the very tiny particles, very small things with the help of light. So it is known as light microscope. If we see anything through the single cell, then single lens, then it is known as the simple microscope. If we utilize the two lenses, then it is the compound microscope. But later on, Knoll and Ruska discovered a very efficient microscope, and that is the electron microscope. The electron microscope this slide is the electron microscope. But where it differ from the compound microscope? In this electron microscope, we observe anything with great magnification, lakhs times magnification. So very small thing which cannot be seen through the compound microscope clearly, it can be seen through the electron microscope. The electron microscope, in this, for observing any object, electrons are utilized. You know the electrons, the negatively charged particles, the fundamental particles of any atom. But the demerit with this electron microscope is that, in spite of the large magnification, it is not able to observe any living thing. As soon as the electrons fall on the cell of the living things, the cell dies. But it is very important in observing anything, very small thing with great magnification, and it enriches our knowledge for studying the very small cell organelles found in the cell. Now, the cell have three basic structure. The first one, the cell membrane, the outermost boundary of the cell. Inside the cell membrane, the cytoplasm, the fluid-like substance present inside the cell membrane, that is the cytoplasm. In cytoplasm, a number of organelles found, small compartments found. Among them, nucleus is very important. Nucleus is the center of the cell. So three things, nucleus, cytoplasm, and cell membrane, all together constitute the cell. The contribution of Purkinje, Huxley, Sladen and Swan, Rudolf Virchow cannot be ignored. Sladen, Swan, and Rudolf Virchow, they proposed or propounded cell theory. The cell theory developed by these scientists, very important one. Sladen, Swan, and Virchow they explain all living organisms, whether plants or animals, are made of cells. A cell is the smallest unit of all the living organisms. And cells develop from the pre-existing cell after cell division. So very important findings given by the Schleiden, Swan and Vercoe. M.J. Schleiden in 1839, a German zoologist, and T. Swan, Theodor Swan, after studying plants and animals independently, put forward a theory called the cell theory. And later on, Rudolf Virchow had the cells made from the pre-existing cell. So different contribution given by the Schleiden, Swan, Virchow. Now the protoplasm doctrine. The term protoplasm was given by J.E. Purkinje in 1840. What is protoplasm? Pro means primitive. So primitive type of the substance having living nature. The term this protoplasm given by Purkinje, the protoplasm means the cytoplasm, nucleus, all together, or the nucleoplasm, the substance found inside the nucleus, together known as protoplasm. Huxley also given its contribution and gave very famous slogan for the cell. The protoplasm is the physical basis of life. Although the protoplasm is a very complex substance, it might be found in the two states, solid state and gel state. When we study the cell, we will find the cytoplasm also differentiated in two zones, distinct zones. The outer and the inner zone. The outer, transparent, clean, non-granular zone, and that is the solid state. It is more liquid-like substance, while the inner cytoplasm, the gel state, more solid-like or granular substance present there. The protoplasm, where different biological processes taking place. The protoplasm always moves in a circular motion. This circular motion, also known as a streaming movement. So, a streaming movement of the protoplasm known as cyclosis. The streaming movement of the cytoplasm known as cyclosis. What is cyclosis? The cyclic movement, the circular movement, the streaming movement of the cytoplasm. Now, slowly, we come to the main points of the cell. The size of the cell varies from organism to organism. Very small size mycoplasma, that is also known as PPLO, pleuropneumonia-like organism. Very small size. It is 0.1 millimicron in diameter. So very small size. Mycoplasma, very specific type of the organism, and at this stage you might understand it is just the smallest bacteria, but later on we will study, or you study in detail, the mycoplasma. And the largest cell, that is the egg of ostrich, and its diameter is 15 centimeters. Very large size. Even the common egg you eat, you buy in the market, that is also a single cell. So this large size cell you can see to the naked eye, but most of the cells, if you want to see, then you must use microscope. So microscopic, but the largest cell found in our body, that is the neuron cell. Different shape and size of the cells are found in different organisms. Here is the diagram of paramecium, guard cell, chlamydomonas, amoeba. Amoeba, you know, most commonly studied organism, unicellular organism, having irregular shape with the pseudopodia on their surface for capturing the food. Nucleus also present in the cytoplasm of the amoeba. The outermost boundary is cell membrane. A number of full vacuoles found, and the contractile vacuole found in this organism. Contractile vacuole, it changes its shape when the water enters into the body of amoeba. The size of the contractile vacuole increases, and when it attains the maximum size, then it shifted towards the periphery and it ruptures, and the excess water of the cytoplasm is removed out, so help in osmoregulation. Neuron cell, longest cell in our body. A number of organs are known, having different shape, size of the cell. One by one, we have to study each part, the plasma membrane or the cell membrane. The cell membrane is a lipoprotein structure, and its functions depends upon the position where it is found. Different cell organelles also surrounded by the cell membrane inside the cytoplasm. It maintains the shape of the cell. It provides mechanical support for the cell. It is semi-permeable, means it does not allow everything to cross. It is selective permeable. It allows only the certain things to cross the cell membrane. So semi-permeable nature of the cell membrane, very important one. When we study the cell, we will study the plant cell, animal cell. In plant cell, an extra covering is formed around the cell membrane, and that is the cell wall. Cell wall generally made up of cellulose that gives rigidity to the plant cell. It is permeable, freely permeable. It allows everything to cross. Non-living cell wall is non-living, but the cell membrane is living. Very important point of difference between the cell membrane and the cell wall. Cell wall only present in the plant cell, and cell membrane found both in plant cell and animal cell. Then come to the cytoplasm. The cytoplasm is the part of the protoplasm. Cytoplasm is the part of the protoplasm. The substance present between the cell membrane and the nuclear membrane, altogether known as cytoplasm. When we study the cytoplasm, a number of inclusions found, a number of things found in the cell cytoplasm. A number of living and non-living structure found in the cell cytoplasm, known as cytoplasmic inclusions. So on the basis of the living and non-living nature, the cell organelles are the living protoplasmic inclusions, while the non-living structures are known as deutoplasmic or ergastic body. Sometimes also we utilize the term cell inclusions for the non-living substance. But the cell organelles are the living. So all cell organelles are surrounded by, most of the cell organelles surrounded by the cell membrane. Certain cell organelles like the mitochondria, chloroplast found in the plant cell, but not in animal cell. While mitochondria found in both the type of cells. So cell organelles are the smaller compartments found inside the cytoplasm and do a very specific job, do a very specific work. As for example, mitochondria, commonly known as powerhouse of the cell. You know, any sort of the system requires energy for working. If there will be no energy, then no system works. Whether it is the non-living system or the living system. Even the computer non-living, it works with the help of energy. So it requires energy. And the different living organisms automatically also it requires the energy. But how it is supplied? So energy produced by the mitochondria. And mitochondria, very important cell organelle, powerhouse of the cell. It is double membrane bound cell organelles. The outer cell membrane or outer membrane, the inner membrane, having large number of the invaginations, finger-like projections. When we study the mitochondria in detail, you will find the invaginations or the finger-like projections known as cristae that possess oxysomes or the elementary particles or fundamental, sometimes it is also known as fundamental particles, but the main name is oxysomes containing the different enzymes for the Krebs cycle, very complex process through which energy is produced. Energy is produced in the form of ATP, adenosine triphosphate. In our body, inside the cell, the energy remains stored in the form of ATP, adenosine triphosphate. So main function of the mitochondria is to produce energy. In addition to mitochondria, Golgi complex, endoplasmic reticulum, ribosomes, and so on. Here in plant cell, you know the chloroplast. The chloroplast is a type of plastid in which the chlorophylls project, and it plays a very important role in the process of photosynthesis. It is found only in the plant cell. So one by one, we study all such things, cell organelles. We can understand the plant cell, animal cell by comparing it. So we must know the difference between the plant cell and the animal cell. Next slide, you can see here, given endoplasmic reticulum, ribosomes, mitochondria, Golgi complex. Endoplasmic reticulum, from the name it is clear, a network of the tubules found inside the cytoplasm for easy transport of the substances from one part of the cell to the another. Ribosomes, that is the center of protein synthesis. Ribosomes are the cell organelle which is not surrounded by the cell membrane. Without membrane, it's a very specific one. Generally, ribosomes appears to be granular, made up of two units, two subunits, smaller unit and the larger. During protein synthesis, they form group of the ribosomes known as polysomes. Poly means many. When many ribosomes come together, attached with the messenger RNA for the synthesis of protein, then they form the polysomes. So ribosomes, very important for the synthesis of different types of the proteins. Sometimes the ribosomes remain attached with the mitochondria, attached with the endoplasmic reticulum and gives the rough appearance. So known as rough endoplasmic reticulum. But some of the endoplasmic reticulums are free from ribosomes and known as a smooth endoplasmic reticulum. Golgi complex. The main function of the Golgi complex is the secretion of the different substances. And in addition to secretion, a number of processes, also modification of the different proteins, addition of certain substance, also done by the Golgi complex. It is known as a complex because it is made up of three types of the components: vesicles, tubules, cisternae, and so on. So today we just introduced the cell. Cells are the structural and functional unit of the any living organism. Cell possesses the characteristics of the living beings, like it also so the process of respiration, multiplication, that is reproduction, and many living characteristics. And the different organisms, like the unicellular organism, made up of only one cell, doing all the process of the living being, like the bacteria, like the paramecium. But here the bacteria are different from the paramecium amoeba because bacteria is prokaryotic in nature, means their nucleus is not distinct, not organized. A very primitive type of the nuclear material found in that cell, bacterial cell, and it comes under the prokaryotic cell. But we are here discussing the true cell, that is the eukaryotic cell, which possesses the nucleus, distinct nucleus surrounded by the nuclear membrane. It contains the nuclear material. In plant cell, there is no centrosome, but in animal cell, the centrosome found that help in the cell division. So plant cell, animal cell, prokaryotic cell, eukaryotic cell, all these terms you have to study. Cytoplasm, cell membrane, chloroplast, endoplasmic reticulum, ribosomes, polysomes. These are the different aspects of the cell, and later on we will discuss the nucleus. Today I am just ending this class.